Okay, so welcome back to this next video in which we are discussing the PI3 kinase AKT mTOR pathway. Okay, so we're currently discussing the PI3 kinase enzymes, and we've seen that there are these three main different types of PI3 kinase enzymes which all differ because they have different substrate specificities. Okay, so we are particularly interested in the type 1 PI3 kinase enzymes, which phosphorylate phosphatidylanocetyl 4,5-bisphosphate molecules, which is a phospholipid molecule that you have a little bit of within the cell membranes. Okay, 2 phosphatidylanocetyl 3,4,5 trisphosphate molecules, which is a phospholipid that you have none of in the phospholipid bilayer of the cell membrane. Okay, and we'll come to why that is later. Okay, right, so we now want to study type 1 PI3 kinases in more detail because there is not just one type 1 PI3 kinase enzyme. Okay, so let's talk firstly about the structure then of PI3 kinase enzymes, and we're going to do this in a very sort of basic way at first, and then we'll make this more complicated as we go along. Right, so first off, PI3 kinase enzymes are not just one protein. Okay, they are actually dimers of 2 proteins stuck together. Okay, there is a regulatory subunit here, which I'll just put as R. So this is going to be the regulatory subunit. Now this is not the portion that actually is going to contain a catalytic domain, which is going to phosphorylate the PI3K into PI3K. Okay, that's going to be in the second portion, the catalytic portion. Okay, but the regulatory subunit is going to be important in controlling the function of the catalytic subunit, which is here. Okay, so all PI3 kinase type 1 enzymes are going to be these heterodimers, therefore, consisting of a regulatory subunit and a catalytic subunit. So let's colour this in. We'll colour in the regulatory subunit in orange here. Okay, so this is the regulatory subunit. And then we'll colour in the catalytic subunit in green here. Okay, right. So we'll start with the different catalytic subunits of PI3 kinase type 1 enzymes, okay, because there is not just one catalytic subunit, there are 4 different catalytic subunits that you can use to build a type 1 PI3 kinase enzyme. Okay, so the 4 catalytic subunits then are called P110 alpha, P110 beta, P110 gamma, and P110 delta. Okay, so these are the 4 different catalytic subunits that you can use to build a type 1 PI3 kinase enzyme. So there are 4 different proteins that you can use in this slot here. Okay, now the whole type 1 PI3 kinase enzyme is then named after which of these 4 catalytic subunits you use in that slot there. Okay, so if you use P110 alpha as the catalytic subunit here, then the whole enzyme, the whole dimer here, okay, and it's called a heterodimer because you've got 2 different subunits dimerized together. Okay, the whole enzyme is then called a type 1 PI3 kinase alpha. Now often people will drop the type 1 and they'll just call it PI3 kinase alpha. So if you don't specify which type of PI3 kinase you mean, people will assume that you mean type 1 PI3 kinase. Okay, if you're talking about a type 2 or a type 3, which very rarely people are, you should specify that it is a type 2 or a type 3. Okay, but if it's a type 1, you can get away with dropping the type 1. So you'd call a type 1 PI3 kinase enzyme, which has as its catalytic subunit, and I haven't told you what its regulatory subunit is, it doesn't matter as far as its naming is concerned, you'd call it by its catalytic subunit, so you'd call it PI3 kinase alpha. Similarly, for the beta, the gamma, and the delta, it's the same, you'd call them PI3 kinase beta, and then onwards, PI3 kinase gamma and PI3 kinase delta. Okay, so this is the naming of the different type 1 PI3 kinase enzymes. Okay, so if you've got P110 alpha as your catalytic subunit, you are then called a PI3 kinase alpha. If you've got P110 beta as your catalytic subunit, you are called PI3 kinase beta. If you've got a P110 gamma subunit as your catalytic subunit, you're called a PI3 kinase gamma enzyme. And if you've got P110 delta as your catalytic subunit, you're called PI3 kinase delta. Okay, now, these type 1 PI3 kinases are further divided into two categories. Okay, so there are type 1A PI3 kinase enzymes, and then there are type 1B PI3 kinase enzymes. Okay, now, PI3 kinase alpha, beta, and delta are type 1A PI3 kinase enzymes. So PI3 kinase alpha, beta, and delta, which means PI3 kinase enzymes of this type 1, which have got as their catalytic subunit either P110 alpha, P110 beta, or P110 delta. Those are all considered as type 1A PI3 kinase enzymes. Okay, then finally, PI3 kinase gamma enzymes are considered type 1B PI3 kinase enzymes. Now, what's the difference between them? Well, the difference is in what can actually activate these two types of PI3 kinase enzymes. So the PI3 kinase type 1A enzymes are going to be activated by receptor tyrosine kinases. Okay, so these ones are going to be activated by RTKs, which we're looking at here. Okay, whereas the type 1B PI3 kinase enzymes, these are going to be activated by the beta-gamma subunits of heterotrimeric G proteins. Okay, now we're not going to discuss those here, but the interesting thing is that the G-beta-gamma subunit of heterotrimeric G proteins can activate a PI3 kinase enzyme, which will activate the exact same pathway as we are talking about, which is being activated through receptor tyrosine kinases. So it's not just receptor tyrosine kinases that can activate the PI3 kinase AKT mTOR pathway. G protein coupled receptors can do it as well. But we're going to be interested in these three because we're talking about receptor tyrosine kinases. Now, how do receptor tyrosine kinases and G-beta gamma subunits activate the PI3 kinase enzymes? Well, they bind to the regulatory subunits here. Okay, so that means that these type 1A PI3 kinase enzymes must have different regulatory subunits to the type 1B PI3 kinase enzymes. Otherwise, how would these ones be regulated by receptor tyrosine kinases, whilst these are regulated by G-beta gamma subunits? Okay, right, so we now want to discuss the regulatory subunits of these type 1 PI3 kinase enzymes. So let's begin with the regulatory subunits of the type 1A PI3 kinase enzymes. So, if you are a type 1A PI3 kinase enzyme, I'll remind you that means that your catalytic subunit is either P110 alpha, P110 beta, or P110 delta. And remember, the enzyme is named purely on the basis of its catalytic subunit. Okay, we don't care what the regulatory subunit is. However, there are actually 5 different regulatory subunits that you can bind onto any one of these catalytic subunits in the type 1A form. Okay, and these are known as P85 type regulatory subunits of PI3 kinase enzymes. Okay, so what are these 5 different regulatory subunits then? Well, there is P50 alpha, then there is P55 alpha, P55 gamma, okay, and then finally P85 alpha and P85 beta. Okay, so these are 5 different proteins which can all function as the regulatory subunit of our type 1 PI3 kinase enzyme, and they are going to be within type 1A PI3 kinase enzyme specifically. So these will associate with either a P110 alpha, a P110 beta, and a P110 delta. And for the naming of the PI3 kinase enzyme, it does not matter which regulatory subunit you have. The name is just based on which catalytic subunit you use, which is why as far as the names are concerned, there's only 3 different types of type 1A PI3 kinase enzymes, because the name just reflects which catalytic subunit you have. It doesn't tell you which regulatory subunit you have. So in fact, under each one of these, you have 5 different forms because they could all be associated with these different regulatory subunits. Okay, but all of these P85 type regulatory subunits are going to be activated by receptor tyrosine kinases. They're going to bind to receptor tyrosine kinases, and we will come back to that in a moment. Okay, right. Meanwhile, just to complete the story, we're not going to study the type 1b PI3 kinase enzymes any further, but I would just like to complete the story of what the regulatory subunits for those are. Okay, so there are 2 different regulatory subunits that the P110 gamma catalytic subunit can associate with, and these are P84 and also P101. Okay, so these are the regulatory subunits that can associate with P110 gamma, okay, to form a PI3 kinase gamma enzyme. Okay, but as I say, those will interact with G-beta gamma subunits of heterotrimeric G proteins rather than receptor tyrosine kinases, and we're specifically looking at this PI3 kinase AKT mTOR pathway, which has been activated by receptor tyrosine kinases, so we're not going to study the type 1b PI3 kinase enzymes any further. Okay, so now a more in-depth study then of these type 1a PI3 kinases, which remember consist of a catalytic subunit, which is either P110 alpha, P110 beta, or P110 delta, and then you have your choice of 5 different regulatory subunits here to associate that catalytic subunit with, okay, to make your full PI3 kinase enzyme. But remember, the naming just depends on which catalytic subunit you have, and not which regulatory subunit you use. Okay, right. So now let's have a more in-depth study of the structure then of these type 1a PI3 kinase enzymes. Okay, so let's start off by studying the structure of the regulatory subunit first. Okay, so remember, we are talking about these P85 type regulatory subunits now, which associate with our type 1a catalytic subunits. Okay, and remember there were 5 different P85 type regulatory subunits. There was P50 alpha, okay, there was P55 alpha, there was P55 gamma, okay, and then P85 alpha and P85 beta. Okay, so we're now going to have a look at what the primary structure of these 5 different regulatory subunits looks like. Okay, so they are single polypeptides, so they're all going to have an amino terminus and a carboxylic acid terminus. So the first thing to say is that these 3 here, the P50, P55s, these 3 here, okay, these are going to be shorter than the P85, so this represents the molecular weight of these things, okay, and these 2 down here, P85 alpha and P85 beta, these are going to have additional domains that these 3 have lost basically by being shorter. So this one's the shortest of them all, but these 2, which are slightly longer than P50, these 2 don't have any really important domains that this one doesn't, okay? So as far as we're concerned, we're just going to look at the domain structure rather than looking at amino acid by amino acid. These ones have the same domains, and these ones have a few extra domains. Okay, so we'll start off by drawing the structure of one of these longer ones, and then I'll point out the bits that these shorter ones wouldn't have. Okay, so here's the amino terminus then of our polypeptide. Okay, now in the case of P85 alpha and P85 beta, you have a domain known as an SH3 domain here. Okay, so this stands for Sark homology domain 3, or Sark homology 3 domain. Okay, so this is a special domain that you find in a huge number of different proteins, and was first identified in the Sark protein once again, which remember is this intracellular cytoplasmic tyrosine kinase enzyme that's just floating around free in the cytoplasm. Okay, then you have another domain that you find in many different proteins, which is called a BH domain. Now, the significance of these two domains is not really understood, because after all, these two domains are not going to be present in the other three, and these three are perfectly functional without them. Okay, so these are just two domains which we find in these longer P85 alpha and P85 beta regulatory subunits. Okay, so what does a BH domain stand for? Well, this stands for a breakpoint cluster homology domain. Okay, so the B is for breakpoint, and then there's also a cluster there that doesn't get a letter, and then the H is for homology. Okay, so this is a BH domain, or a breakpoint cluster homology domain. Right, after that, what you have is some of the important domains coming now. So these are the ones that are going to be present in all of these P85 type regulatory subunits. So these two that we have just talked about here, these are only present in the P85 alpha and the P85 beta, these longer ones. Whereas what we're about to talk about, the bits that start now, these are going to be present in all of the 5 of them. Okay, so these other 5, if you like, would start now. Okay, so the first really important domain then is going to be what's known as the N-Terminal Sh2 Domain. Okay, so SH2 domains, remember, are these important domains which are capable of binding to phosphotyrosine residues. And indeed, this SH2 domain is going to be extremely important for recruiting the regulatory domain to a phosphotyrosine domain on the intracellular aspect of the receptor tyrosine kinase dimer. Okay, so this is the N-SH2 domain, because we're going to have another one which will be closer to the C terminus. So this is the SH2 domain, the Sark homology 2 domain, which is closer to the amino terminus of the protein. Okay, and it's closer relative to the other one, which we'll come across later. Okay, then you have a special region known as the inter-SH2 region. Okay, so this is called the I-SH2. Now, this is not an SH2 domain. This is just the domain that's in between the two separate SH2 domains. Okay, so I'll colour this in yellow. This is the I-SH2 domain here. And this is going to be important in binding to the catalytic subunit. So I need to be careful not to obscure the position where I want to draw the catalytic subunit. So this is called the inter-SH2 domain. Okay, so this is just an important portion that's in between two SH2 domains. Okay, after the inter-SH2 domain, you then have the C-SH2 domain, which is the one that's closer to the carboxylic acid tail here. So here's the carboxylic acid tail. So this is the C-SH2 domain because it's closer to the C terminus. This one's the N-SH2 domain because it's the one that's closer to the N terminus. Okay, and then the polypeptide ends with its carboxylic acid terminus here. So these are the interesting domains. Now these two here, these, as I say, are the accessory ones that are added on these longer ones. If we were just looking at a P50 or P55 alpha or a P55 gamma, okay, the amino terminus would be here, and then you'd just be starting the polypeptide, and it would be just these 3 here. And these are the 3 essential ones. Okay, so don't worry about these 2. These are the ones that are going to be interesting. Okay, right then. So this is the regulatory subunit, okay? This is the subunit that's going to be involved in binding to the receptor tyrosine kinases, and it's also involved in binding to the catalytic subunit now. So we want to now study the catalytic subunit, okay? So remember, we are talking about the catalytic subunit of a type 1A receptor tyrosine, PI3 kinase enzyme, okay? So it's either P110 alpha, P110 beta, or P110 delta. Now, once again, all 3 of these proteins are single polypeptides, so we can start with the amino terminus and then work our way down to the carboxylic acid terminus. Okay, so we'll start with the amino terminus here. And the first really important domain is going to be the domain which binds to this inter-SH2 domain, and this is how the regulatory domain associates with the catalytic domain here. Okay, so this is the ABD domain here. Okay, and this stands for adapter binding domain here. Okay, so the A is for adapter and the BD is for binding domain. Now, why is it called that? Well, if you think about what the regulatory subunit is going to do, the regulatory subunit is going to bind to the phosphotyrosine residues of the receptor tyrosine kinase dimer via its NSH2 domain and its CSH2 domain. It's also then going to bind to the catalytic subunit via its inter-SH2 domain here. So effectively, it is just functioning as an adapter. It's a protein whose job it is to bind to the receptor tyrosine kinase and then bind to the catalytic subunit, which is the target of the receptor tyrosine kinase. So it's just an adapter protein sitting in between those two interacting proteins. Okay, so that's why this is called the adapter binding domain, because it's the domain which is going to bind to this adapter protein here. Okay, so here it is, the ABD. Now, after the ABD, there is then a region which we're not going to talk about hugely, okay, which is called the RAS binding domain, the RBD. Okay, and that relates to the fact that RAS monomeric G proteins can also activate these catalytic subunits by binding here. Okay, but we're not going to dwell on that. Okay, we're going to focus on the activation by the receptor tyrosine kinase itself. So this RBD domain, this is a domain which can be bound to by RAS monomeric G proteins. RAS binding domain. Okay, and they can bind directly to the catalytic subunit, so they don't need an adapter protein sitting in between them, and they can activate and recruit these catalytic subunits. Okay, but as I say, we're not going to dwell on this point here. Okay, so this is in turquoise here. Okay, then after the RAS binding domain, you have another domain known as a C2 domain, which is a domain that's important in calcium signaling usually. Okay, so usually what can happen to C2 domains is that they bind to calcium. Okay, and this is important, for instance, in enzymes such as protein kinase C enzymes, which all have C2 domains, and the calcium helps to activate the protein kinase C enzyme. Okay, it's not going to play a huge part in our story here. Okay, but it's an interesting domain nevertheless that's there. Okay, then you then have a domain that's known as the helical domain, and then following the helical domain, you then have a portion that is actually responsible for the phosphorylation of the PIP2 molecules into PIP3 molecules, and this is the catalytic domain, so I'll draw it in the classical way to draw an enzyme. Okay, so this domain here, this is the portion that actually carries out the reaction. So this is the catalytic domain of the catalytic subunit of our PI3 kinase enzyme. Okay, so let's colour in the helical portion in pink, and then we'll have the catalytic domain here in orange. Okay, right, so at present, this is our inactive PI3 kinase enzyme of the type 1A. Okay, so it's got one of these P85 type regulatory subunits here, and bound to that we've got either P110 alpha, P110 beta, or P110 delta. Okay, what is now going to happen is this thing is sitting within the cytoplasm when it's inactive, so it's just floating around the cytoplasm. What's now going to happen is when we get our receptor tyrosine kinase dimer forming, that receptor tyrosine kinase will have these phosphotyrosine residues on its intracellular aspect, and those phosphotyrosine residues are going to recruit the whole structure up to the underside of the phospholipid bilayer, and they're also going to produce conformational changes that then lead to the activation of the catalytic domain here. Okay, so let's go through this. So, let's have our phospholipid bilayer here. So let's just redraw out our receptor tyrosine kinase dimer again, because it's been quite a while since we saw it last. Okay, so here, remember, is our ligand binding domain with the amino terminus extracellularly here. Then we have our membrane spanning alpha helix here. Remember, our ligand binding domain has bound its ligand, and we're imagining that this is the epidermal growth factor receptor here, so the ligand would be epidermal growth factor. Okay, so here is a ligand molecule bound to this receptor tyrosine kinase's ligand binding domain as well. Okay, so we'll colour in both of these ligand binding domains, which are dimerized together, in orange. Then we'll colour in the ligand, which is the epidermal growth factor, in turquoise. Then we have our membrane spanning alpha helix here. I think we'll colour that in red. Okay. And then on the intracellular aspect, we have our tyrosine kinase enzyme here. And I'm going to extend the cytoplasmic tails a little bit so I've got more room to play with. Okay, so then we've got our tyrosine kinase over there as well. Here's our carboxylic acid terminus, and here's our carboxylic acid terminus of this other receptor tyrosine kinase. Let's colour both of the tyrosine kinase domains in blue here. And then, of course, once this is dimerized, what then happens is one of these phosphorylates the other, okay, very slowly, because remember, before they've been phosphorylated, they're not very active. That activates this one, and now it will phosphorylate the other one, okay. Now they're both active, and what they're going to do is phosphorylate the cytoplasmic tails of the opposite receptor tyrosine kinase. So this one here will phosphorylate tyrosine residues on this receptor tyrosine kinase's cytoplasmic tail, and this tyrosine kinase domain here will phosphorylate tyrosine residues on this one's cytoplasmic tail, like so. Okay, so here are our phosphotyrosine residues. Now, what's now going to happen is both the N-Sh2 domain and the C-Sh2 domain are going to come and bind to phosphotyrosine residues on the intracellular aspect of our receptor tyrosine kinase. Now, to try and simplify the picture down, I'm going to get rid of showing the SH3 domain and the BH domain. Okay, they're only present in the P85 alpha and the P85 beta. Okay, so we'll imagine we're working with one of these smaller ones. We'll just show the essential domains, okay, just to try and simplify the picture down. Okay, so here then, let's have our NSH2 domain here, binding to a phosphotyrosine residue. Okay, so here now is the amino terminus of our regulatory subunit of our type 1a PI3 kinase enzyme. Okay, now I'm not going to try and draw NSH2 in there, I'll label it up instead. So this is our amino terminal SH2 domain here. Okay, so I'll colour this in red, I'll keep the colour coding the same. Okay, so this is now the first important domain because we're imagining we're dealing with one of these smaller ones that doesn't have these other two domains here that aren't really characterized that well. Okay, right. Then we've got this inter-SH2 domain here, okay, which I'll colour in yellow. Now that's not binding to phosphotyrosine residues, so don't think that that's an SH2 domain, it's the inter-SH2 domain, remember. And now we've got the CSH2 domain, which will be binding to a phosphotyrosine residue over there. Okay, and we've previously shown that up in pink there. So we'll do that again. So here it is, this is the CSH2 domain. Okay, and I think since all the others are coloured fully in, I'll keep that one the same. So this is the CSH2 domain. Okay, and that's now the end of our regulatory subunit here. Okay, so there's its carboxylic acid tail. So it's bound to this phosphotyrosine residue and this one here. Okay, now of course it's still bound to the catalytic subunit here. Okay, so here's the adapter binding domain, and here's the amino terminus of our catalytic subunit of our type 1a PI3 kinase enzyme. Okay, so let's colour in this adapter binding domain in vivid purple again, and label it up as well. So this is the ABD, or the adapter binding domain. Okay, then after the adapter binding domain, you have that RAS binding domain, okay, which I'll have here. So this is the RBD, the RAS binding domain, okay, which we're not going to talk about. Okay, after that you've got this C2 domain, which again isn't going to play a very important part, and then after that the helical domain. Okay, and then the really important portion, which is the catalytic portion. Okay, so we'll have our catalytic portion now perfectly in position to work on the PIP2 molecules, which are at the which are in the inner leaflet of the phospholipid bilayer. Okay, so that's the first function of this binding to the phosphotyrosine residues on the intracellular aspect of the receptor tyrosine kinase dimer, that we have just recruited the whole PI3 kinase up to the underside of the phospholipid bilayer. Okay, so let's colour in some of these other domains. So here is the C2 domain in blue here. Okay, here's the helical domain in pink. And then here now is our catalytic domain in orange here. Okay, so we've now recruited our enzyme to the underside of the phospholipid bilayer. Now, not only that, and that's a really important part, in fact I should stress more about how important just the mere recruitment of the whole thing to the underside of the phospholipid bilayer is. Okay, this catalytic domain is not completely inactive. Even when it's floating around in the cytoplasm, it is capable of phosphorylating PIP2 molecules. Okay, it's just it can't find any because, you know, it's floating around in the cytoplasm. It's not going to find any there. They're all in the cell membrane. Okay, so if you just recruit it and put it underneath the phospholipid bilayer like this is enough to get it doing its job, basically, get it converting PIP2 molecules into PIP3 molecules. However, the receptor tyrosine kinases generally do more than that. Generally, they also cause conformational changes that then lead to the further activation of the catalytic domain. So the receptor tyrosine kinase does both of the things that it can do. It relocalizes the enzyme to the underside of the phospholipid bilayer, and it also causes a conformational change that then activates the enzyme. So this catalytic domain here then now is going to start converting phosphatidylinosytyl 4,5-bisphosphate molecules into phosphatidylinosytyl 3,4,5-bisphosphate molecules. Okay, so in the membrane, you're going to get the conversion of PIP2 molecules into PIP3 molecules. Okay, right, so let's show this a little bit further down here. Okay, right, so if this is the membrane, I'll now actually show a PIP2 molecule sitting within the membrane. So here are the two long-chain carboxylic acids. Here's the phosphate group coming off the alcohol group of the third carbon of the glycerol molecule. Here's our inositol ring with the two phosphate groups coming off it. Okay, so here are the phosphate groups in pink. Here is the inositol ring in blue. In green we've got the glycerol molecule, and then we've got the two long-chain carboxylic acids which are attached here. Now the catalytic domain is going to convert this into PIP3, so let's draw this here. Okay, so once again there's the phosphatidic acid molecule, here is the inositol ring, and now I've got phosphate groups coming off the third, the fourth and the fifth carbons of the inositol ring. Okay, so let's colour all of these phosphate groups in purple here. Okay, then we'll colour in the inositol ring in blue, then the glycerol molecule in green here, and then the long-chain carboxylic acids in orange. Okay, right, so this is what is occurring once we recruit and activate the PI3 kinase type 1 enzyme to the underside of the phospholipid bilayer like so. Okay, so I said that we have PIP2 as a part of the cell membrane, okay, so it's a normal member that you find within the cell membrane. It's not particularly high concentration, but you do have it there, whereas you don't have that much PIP3 at all. And the reason is that there are enzymes whose sole job it seems to be to convert PIP3 back to PIP2, to chop this phosphate group off, and the most famous example is P10. Okay, so P10 stands for phosphatase, okay, that's the P, and then the 10 stands for and-tensin homolog, so phosphatase and tensin homolog, so I'll put in the and as well, and-tensin homolog. Okay, so these enzymes scour the cell membrane looking for PIP3 molecules, and when they find them, they cut off that phosphate group and return them back to PIP2 molecules. So this is the reason that usually there is no PIP3 in the cell membrane. Okay, so when we recruit PI3 kinase enzymes of the type 1A to the underside of the phospholipid bilayer like this and activate them, we're going to get the production of PIP3 from PIP2, and therefore or transiently we're going to get the appearance of PIP3 molecules in the inner leaflet of the phospholipid bilayer, and it's the appearance of these PIP3 molecules that are then going to activate downstream signaling molecules, basically. And we'll turn our attention to those downstream signaling molecules in the next video.